This Embryology Highlights screencast, Early Embryogenesis 1, deals with fertilization, cleavage, gastrulation, and notochord formation. An embryo begins as a single-cell zygote formed when a sperm gamete fertilizes an oocyte gamete. The large zygote undergoes a series of cell divisions to produce normal-sized cells. This process is called cleavage. It's the process of cell division without cytoplasmic growth. Cleavage produces a blastula or blastocyst. The next step is a gastrulation, by which the blastula develops 3 germ layers. These 3 germ layers, ectoderm, mesoderm, and endoderm, give rise to all of the organs of the body. In mammals, the process of organogenesis takes place the period of the embryo, and that's followed by a fetal period when the organs grow in size, leading to birth and then eventually an adult individual. So what we see is a process by which one cell eventually becomes billions of cells. So mitosis produces genetically identical cells. They differentiate into different roles by virtue of local influences that turn some genes on and some genes off. On the other hand, gamete formation, the formation of the oocyte and the spermatozoon, that cell division process called meiosis, and meiosis produces genetically different offspring cells. And so each gamete is genetically different, whereas each cell produced by mitosis is genetically the same. All of these cells, of course, are diploid. Gametes are haploid. And so meiosis, in addition to producing genetically different offspring, it produces haploid offspring, half the number of chromosomes found in a normal diploid cell. Fertilization takes place in the uterine tube. An ovulated oocyte meets sperm. One of them will fertilize it. Early cell divisions take place in the uterine tube as well. Oocyte and the early cell divisions are surrounded by the zona pellucida, which prevents implantation. Fertilization involves the union of a haploid oocyte and a haploid spermatozoon. That union produces a diploid zygote. That zygote starts to divide, and that's the beginning of cleavage and the end of fertilization. This is an ovulated oocyte. It, along with some surrounding follicle cells, granulosa cells, were ovulated. You can see that the oocyte is very large compared to the normal-sized granulosa cells. The oocyte is surrounded by a zona pellucida, a glycoprotein membrane that it secretes. Fertilization begins with a sperm cell weaving through these granulosa cells and binding to the zona pellucida. There are receptors on the zona pellucida that are recognized by the sperm cell, the mechanism for making sure that the same species sperm fertilizes this oocyte. Sperm, after it makes contact with the zona pellucida, has to release enzymes. The process is called acrosomal reaction. And those enzymes then denature the zona pellucida and allow the sperm cell to work through the zona pellucida to make contact with the cell membrane of the oocyte. Sperm will fuse with the oocyte. That triggers the oocyte to actually complete meiosis. The union of the sperm with the oocyte triggers the oocyte to depolarize its plasma membrane by extruding calcium. It also causes it to release enzymes from vesicles that are along the surface that denatures the zona pellucida to prevent other sperm from also fertilizing it. Polyspermia would be a fatal condition. Fertilization ends with the 2 nuclei coming together and beginning mitosis. This image illustrates cleavage. We have drawings of cell divisions. Down below we have actual embryos of a 2-cell stage and a multiple cell stage and many cell stage. This is the zygote following fertilization. The sperm nucleus and the oocyte nucleus then are going to begin a mitosis. We have 2 cells, Each of those is going to undergo a mitosis, so 4 cells and then 8 cells and so on. Notice that the cells are increasing in number but not increasing in cytoplasm, so that this is what cleavage is. There's a cell division but not a cytoplasmic growth. And so we're ending up with an end product of many normal-sized cells. Meanwhile, all of this is taking place initially within the zona pellucida until this cell mass gets so large that the zona pellucida becomes fragmented. This shows that cleavage leads to the formation of a morula and a blastula. So a morula is a ball of cells within the zona pellucida. A blastula or blastocyst contains a hollow cavity, fluid-filled cavity, called a blastocele. We differentiate then an outer boundary formed by trophoblasts and inner cells called an inner cell mass. The cells that are produced during cleavage are called blastomeres. So each of these, this is a blastomere. Initially, these are all surrounded by a zona pellucida which prevents implantation. But as the blastocyst grows and enlarges, the zona pellucida is discarded, and this is the stage that's suitable for implantation. These early cells during cleavage are all pluripotential. Each of these early cells can form an individual, a separate embryo. And so what can happen then is that these cells can separate. Each can grow into a separate individual. And that's the explanation for monozygotic or identical twins, as opposed to fraternal twins where there has to be 2 fertilized, and so you have 2 genetically different persons who are sharing the same pregnancy. But in monozygotic twins, identical twins, you really have clones because they come from an early zygote that split at this early stage. Then, when you get to the morula stage, we start to see cell differentiation. It's related to the position of the cells. So the cells that are inside are going to form gap junctions, which favors communication, exchange of information. They will become the inner cell mass that will become the embryo plus the yolk sac and allantois. Meanwhile, the outer cells, the outer blastomeres, differentiate in the sense that they form tight junctions, they become secretory, and we refer to these then as trophoblasts, and they are going to form the chorion and the amnion fetal membranes, and their tight junctions form a seal, and their secretions then create this fluid-filled blastocyst. This image shows stages of blastocyst development. Here is an early blastocyst. It's surrounded by a zona pellucida. You can see inner cell mass and trophoblasts on the outside, and the beginnings of a blastocele. Then we have a later stage where there's been a proliferation of trophoblasts and an enlargement of the blastocele. Zona pellucida is under stress and being discarded. Finally, we have a more mature blastocyst. We have this elongated trophoblast layer and then the inner cell mass. So this is going to become chorion and amnion, and this is going to become the embryo itself plus the yolk sac and allantois. This entire structure, the inner cell mass and then the trophoblast, together constitute a conceptus. So next, the embryo undergoes a process called gastrulation, and that gives rise to the 3 germ layers, ectoderm, mesoderm, and endoderm. They're called germ layers because they give rise to all of the organs of the body. Ectoderm will form the skin and the nervous system. Endoderm will form the lining of the gut and respiratory system and lower urinary tract. And mesoderm forms everything else, particularly skeletal muscle and much of the bone of the body, and circulatory and urinary and genital systems, the gonads and the kidney. The first stages of gastrulation produces a hypoblast layer and a yolk sac. This image shows a low power view on the right and a high power view of the inner cell mass region on the left. And there are three levels, an earlier, intermediate, and then a later stage of development. What we see happening first is delamination of the inner cell mass. That is, it's giving off a cellular lamina called the hypoblast layer. That eventually it forms a complete yolk sac, and it creates a space, a coelom. Looking here then, here we see the hypoblast layer, the yolk sac, and here's the coelom. And here we see the inner cell mass and then the trophoblast layer. Now, in our domestic mammals, what happens is that these trophoblast cells degenerate over the inner cell mass, and that produces this situation here, which we can now call the epiblast. So the future embryo is right at the surface in our domestic mammals. It's different in primates and rodents. What happens is, instead of this degenerating, a space develops between the trophoblasts and the inner cell mass, and that space becomes the amnion. In our domestic mammals, the amnion cannot form in that way, and so it has to form by a folding of the trophoblast to create an amnion, and we'll see that later. So here we have switch views. We're looking at the surface of the embryo. We can say the brown is the trophoblast, and the yellow area here is the inner cell mass. And we're looking at stages of development, and then this is a later stage. Initially, there's just an epiblast. You don't know which is the head, tail, right or left, or anything. And then a series of thickening, a proliferation of cells begins. It progresses, and it develops what we can call a primitive streak. When that occurs, now we know that this is the head end, this is the tail end, this is the right, and this is the left. That primitive streak progresses, and it develops a groove in the center with elevated proliferated cells forming ridges on either side of the groove. So this is a primitive streak. It has a primitive groove surrounded by primitive edges and a primitive node at the cranial end of it. This is an artist's drawing of a primitive streak cut in cross-section. Here it is the same thing colored. Epiblast is in blue here on the surface, and you can see that there's a proliferation of cells on the shoulder. That's the source of cells that are flowing in and filling the coelom between the epiblast and the hypoblast. Down below, we can see arrows that are showing how this flow of cells is occurring. It's from the shoulder, then it's going in and filling up the coelom. That really creates the three germ layers. The first cells that flow in, shown in yellow here, are ones that join the hypoblast layer. They are going to be the lining of the gut, and the hypoblast was going to form the yolk sac, and so you can see that the gut and the yolk sac will always be connected with one another. Cells shown in red are going to be mesoderm, and they're going to give rise to muscle and much of the bone and connective tissue of the body, as well as kidneys and gonad. Meanwhile, the cells that remain on the surface will be the ectoderm, and they'll give rise to skin and nervous system. This drawing shows embryonic cells in two forms. One, which could be called epithelioid, they resemble an epithelium, is where the cells are in contact with one another. That's great for communication and forming barriers or layers. The other form you can see in this flow into the coelom. That form is called the mesenchyme. These cells are not in contact with one another. So the mesenchyme arrangement is great for migration, for flowing, but it's not very good for communicating. The epithelioid is great for communicating, but it's not good for migrating. Embryonic cells will go from a mesenchyme form to an epithelioid form and back again. This first mesenchyme is called the primary mesenchyme. So it's the primary mesenchyme that fills the coelom, comes through the primitive streak, that gives rise to the endoderm, the mesoderm, and then by default the ectoderm. This image shows the 3 germ layers at a later stage of development. The embryo is still flat. We can see ectoderm on the surface. By this time, a nervous system has formed and separated from the ectoderm. We see in yellow here the endoderm, which is forming the lining of the gut. And then between the endoderm and the ectoderm, we have the mesoderm. We recognize medial, intermediate, and lateral mesoderm. The medial mesoderm is going to form somites. The intermediate mesoderm is going to form gonad and kidney. And the lateral mesoderm undergoes a cavitation which reestablishes the coelom. That splits the lateral mesoderm into 2 parts separated by a coelom. The lateral part, called somatic mesoderm, is going to join with the ectoderm, and the medial layer, which is called splanchnic mesoderm, is going to connect with the endoderm. The splanchnic mesoderm and endoderm are going to form the wall of the gut. The somatic mesoderm and ectoderm are going to form the body wall, and there'll be a coelom in between those. Also present here is a notochord, and that's what we want to discuss next. The notochord grows anteriorly from the primitive node. It has a small role in the adult. It ends up being the nucleus propulsus of an invertible disc, but it has a critically important role in the embryo because it induces the head process to form, it induces the nervous system to form, and it induces somites to form, and somites will give rise to skeletal muscle and vertebrae. This image shows the notochord in red, and it shows it in a sagittal view, in a dorsal view, and in a transverse view. And you can see that it grows out from the primitive node. It grows in an anterior direction, and it occupies the space between the endoderm and the ectoderm where primary mesenchyme does not invade. So the mesenchyme comes through the primitive streak and spreads around, but it doesn't invade this midline anterior to the primitive node. And so it's that space where this rod-shaped notochord grows, and so we see it here between the ectoderm and the endoderm. And so this is the notochord, which is going to induce formation of the head process, the nervous system, and somites. This is a later stage embryo. It is no longer flat, it's a tubular embryo, but it shows the effect of the notochord. It has a head process here. Inside the head we have a brain and then a spinal cord caudal to that. And then you see these chunks of tissue, and these are somites. So there's a series of somites along the embryo. The somites, the nervous system, and the head are all induced to form by the notochord. This last image shows a nucleus propulsus of an intervertebral disc. The nucleus propulsus in yellow here is what the notochord ends up becoming as an adult. It's surrounded by an annulus fibrosus of fibrocartilage, and together these make up an intervertebral disc, the mass between bodies of adjacent vertebrae. Here we see the arch of the vertebra, and we see the nervous system within that, and of course this was induced to form by the notochord as well. This concludes this Embryology Highlight screencast, Early Embryogenesis 1.